Everyone, so my name is Iuliana Ionita. I am a faculty at Columbia University, and I'd like to first thank the organizers for putting together such a nice workshop. So I'm going to talk about some work in my group on integrative statistical approaches for the analysis of whole genome sequencing data. So as you all know, it's becoming increasingly more efficient to generate whole genome sequencing data on large number of individuals. And there are a few large-scale projects going on, such as TopMed, that plans to generate whole genome data on more than 120,000 individuals, in addition to other types of omic data like metabolic profiles, protein and RNA expression levels, and so on. Another large-scale project is UK Biobank, and they plan to generate whole genome data on 50,000 individuals in this biobank. And there are some smaller-scale projects. For example, we are involved in an autism sequencing project that will soon have available over 20,000 whole genomes. So with all this whole genome sequencing data becoming available, we need to think about ways to efficiently analyze this type of data. And some of the main difficulties that occur in handling whole genome data have to do just with the nature of the data. So when we sequence whole genomes, we're going to identify large number of variants in the non-coding parts of the genome. For the most part, these variants have low frequency in the population. And furthermore, we have a fairly poor understanding of their functional effects at the molecular level. So it's really difficult to prioritize the variants we identify based on their functional effect. So given all these issues, we have to be a little careful of how we analyze this data. And standard approaches we've been using for analysis of common variants in GWAS will probably not be very efficient in the context of sequence data and rare variant data. And to begin with, as I mentioned, most of the variants we discover in sequencing studies are rare. So if we think about the sample sizes we need to identify association with these variants, they're going to be very large. So what I'm showing here in this plot is sample size as a function of the effect size, relative risk, for different minor allele frequency. So if we look at the black curve, which corresponds to a variant with frequency 1 in 1,000 in the population, if we assume modest effect sizes for these variants, which is probably the case, then we need upwards of hundreds of thousands of samples to be able to identify association with such variants. So obviously, we need to think about better ways to deal with this type of data. Another difference from what we've been doing so far with common variants analysis, so when we look at the region, we will see a small number of common variants. So in this example, we have one common variant. You see it's more common in cases versus control. So it's good. We can test for association at this individual variant and hope to be able to detect the association. However, once we look at rare variants, what we see is that we have a lot more rare variants in this region compared with common variants, and each of these variants is individually rare. So in any given data set, most of the rare variants you'll see a few times, once or twice. So basically it's impossible to detect association one variant at a time, right? But what you can see in this example is that overall, the cases have more of these rare variants compared with controls, which suggests that instead of testing one variant at a time, we could do something different. We can test at the level of the gene. So in the first part of the talk, I will discuss some of the main ideas behind these gene-based or region-based association tests. 5-10 years ago, there was a lot of activity in this area in developing region-based association tests. And mostly it comes down to two classes of tests, the burden test and dispersion or SKAT tests. And then there are a lot of extensions of these basic tests. So there is a very useful extension called SKAT-O, which is an optimal combination of the burden and dispersion test. And then there are integrative tests, the ones that I'm going to talk about, where you integrate functional annotations into the testing procedure to hopefully improve the power of this test. So here are the basic ideas behind this region-based association test. So we assume we have sequenced n individuals in a particular region, say a gene. For each variant, we have a phenotype, could be like a binary or a continuous trait. We have, for each individual, we have a set of covariates. So this could be age, gender, principal components, so anything we want to adjust for. As I said, we sequence a region. So now for each individual, we're going to have a vector of genotypes. So we have p variants, and so we have a vector of p genotypes. And then for each of these variants, we have a set of functional scores. So this could be evolutionary conservation scores or epigenetic scores or predictions of functional effect for these variants across different tissues or cell types in the human body. Okay? So the goal is basically to test for association between Y and G while adjusting for X, and taking into account the information from Z, from the functional scores. Okay? So we consider this classical linear mixed model that relates the phenotype to the genotypes. And so we're interested in testing the null hypothesis that these p regression coefficients are all 0. Okay? And so you could use standard methods. You can test one beta at a time, or you can use multiple degree of freedom tests to do a joint test. But because of the issues I mentioned with the data, you have lots of p is large, you have sparse data, you have low frequency variance, then this type of traditional testing schemes are not powerful. So we have to basically do something a little different, and in particular to improve power, we'll have to make some assumptions on these effects, betas. So what the main idea behind burden and SKAT is basically, so it turns out, what's really nice about these two tests is that, you can put them in, they can be derived within the same framework. So what we're going to do, we're going to assume that beta j are random variables that come from arbitrary distribution with mean 0 and variance wj squared tau. We assume an exchangeable correlation structure among the betas. So correlation between any two betas is equal to rho. And under this assumption, the null hypothesis we had before, testing all betas equal to 0 comes down to testing only one single parameter, tau is equal to 0. And so the burden test is basically, you take rho equal to 1. So all betas are completely correlated. So in other words, all betas are the same up to this weighting factor wj. And so it's called the burden test because what we're actually doing, you're calculating for each individual a burden of a genetic score weighted by these weights, so this, right? And then you regress the phenotype on this weighted genotype score, okay? For the other type of test, dispersion or SKAT, you basically assume that rho is equal to 0. And so the underlying score statistics for these burden and SKAT tests, they can be computed easily, and they have some very nice forms. So when rho is equal to 1, as I said, we get the burden test. And the score statistic is basically the square of this linear combination of individual variant score statistics. So you calculate a score statistic for each individual variant, and then you sum overall variance in the gene or region. When rho is equal to 0, we get the SKAT test, as I said, and basically has this quadratic form. So you take the score statistic for each variant, you square it, and then you do the sum. So you can see that, these different tests perform optimally under different conditions. One easy thing to see is that, if you have a mixture of deleterious and protective variants, the burden test will lose power because you'll have here, plus and minuses that cancel out. Whereas a SKAT test, because it squares the contribution of each variant before you aggregate across variants, is robust to such scenario wherein you have a mixture of risk and protective variants. Okay, so that's why, because they're sort of optimal under different conditions. So there was this test called SKAT-O proposed, which is an optimal combination of the burden and the SKAT square statistic, and it turns out to be like a good choice for one single test, and it performs better than individually burden or SKAT test. For a fixed value of rho, of the correlation among betas, the null distribution of Q is basically a mixture of chi-squares, and you can get p-value analytically. So these tests are very, they have some very good advantages. They're computationally efficient. You can get analytical p-values easily. So they have they're regression-based tests, so they have a lot of good features. I'm sorry?
The row 0 from the last slide.
Oh, so you basically, so what you do is like you search over a grid of values between 0 and 1, and it's like it's data adaptive. So you let the data decide what combination you use. So it's like a minimum. So you basically calculate a p-value for each. So you have a grid of raw values between 0 and 1. And then you calculate the overall statistic is going to be the minimum of those p-values over the grid, and then you basically, that's how the.
So you choose it to be the row 1, which will give you the most significant result.
Yeah, it will. The ones that will give you the minimum p-value. Yeah. So now suppose that we are actually interested in incorporating functional annotation data into the square. So as you can see in the original burden or SKAT, we can only incorporate one WK at a time. So one functional score at a time. Suppose now that we have functional scores across many different we could have like many different tissues or cell types in the human body, or just simply many different functional scores. How can we incorporate those functional scores into the test? So what we are going to do, we're going to partition the genetic effect beta J into different components corresponding to, to these different functional scores. So we're going to write beta J as gamma J0 plus, we sum over Q functional scores, ZJK, this is a functional score for the case functional score for the J's variant times gamma JK. And gamma JK are random variables with variance gamma JK equal tau K, and we assume the same exchangeable correlation structure as before. And so the null hypothesis now is basically to test that all these variance components from tau 0 to tau Q are 0. Okay, so for a given raw correlation between 0 and 1 and a given functional score K, we basically calculate this square statistic Q rho K, which is a linear combination of sort of the burden and the SKAT statistic for using this functional score K. And now, if we look across a large number of values of correlation coefficients, rho, and a large number of functional scores, and we have basically a set of score statistics, and we can calculate, we can get the p-value for each of them, and then our overall test statistic is going to be this mean p statistic. And we've developed an efficient resampling-based moment matching approach to calculate p-values. Okay, so these are sort of the main ideas behind these gene-based tests. I'm going to show some application to a Metabochip data set. So this is a data set on lipid traits. So we have HDL, LDL, total cholesterol and triglycerides for 12,000 individuals. And we focus on 100 fine mapping regions. Within this region, there are a total of 266 genes. So we're going to basically do this gene-based test, and we're going to compare the analysis using SKAT-O, like the standard SKAT-O analysis, with an analysis where we integrate functional scores. And I haven't talked about these functional scores. I'll talk in the second part of the talk. But basically, for each variant, we're going to have a functional score in 127 different tissues. So basically, for each variant, we have a probability that that variant is functional in a tissue. And we have this for 127 different tissues. Okay? So what I'm showing here on the x-axis is the physical position of the genes. And on the y-axis is a negative log 10 p-value from this gene-based test. And as I said, we have two types of tests. So the circles correspond to the integrative test, and then the triangles correspond to the standard SKAT-O test. And so what we basically get is that you'll see that the circles tend to be above the triangles. So basically, by doing this integrative analysis, we are able to get smaller p-values, more significant results than doing a standard SKAT-O analysis. And we've also shown in simulations that if you integrate function annotation scores, even if only like a very small number of those function annotations are correlated with underlying effects like betas for the variants, you can gain increases in power compared to not including those function scores.
Iuliana, do you have any kind of QQ plot or anything? Or I'm sure you looked at this.
Yeah, we'll
make sure there's no inflation in the integrated scores.
No, I think we looked at this. I think the type 1 error is well controlled. Yeah. Mm-hmm. It's really just, when you have so many scores, some of them will be correlated with underlying betas for the variants in a particular gene. And so that kind of helps you a lot in terms of power. Okay, so this is like, we look at, so what I've talked so far is doing this gene-based or region-based test. Now suppose you are looking at whole genome data, and you're in non-coding parts of the genome, so there is no natural functional unit for analysis. So what we're going to do in that case is basically something very natural. We're going to use scan statistics. So the underlying idea in scan statistics is basically to slide a window of a fixed size W along the length of the region or the genome, and compute an overall statistics for all windows of length W. So the challenge is that these windows are correlated. So then the test statistics for adjacent windows will also be correlated. Also, if you include multiple functional scores, that will induce additional correlation. So we need to basically account for this correlation in our analysis. So what we want to do, so we do this sliding window approach, right, across the region. So what we want to do is to identify those windows with p-values less than a certain threshold. And we want this threshold to choose this threshold so that we can control the family-wise error rate at some level, okay? So a simple way to control the family-wise error rate is to use the Bonferroni correction, but that will have, that will be conservative because of the, correlation among the windows. So we're going to try to derive better thresholds that accounts for this correlation. So basically for any windows phi, KL, we can get a p-value. And then we define this extreme value statistic T as the maximum overall windows, and if you use the functional scores, overall functional scores, of the negative log p-value. And then the significance threshold, basically alpha star, can be basically easily derived to have this form. And so what we want, basically, we want to be able to calculate this, the quantiles of this, of the distribution for T, okay? And I'm not going to show you how we do it, but basically we use some results from extreme value statistics theory and some resampling-based approach that accounts for correlation among windows to basically calculate the quantiles of this distribution, and then basically we can get the significance threshold alpha star. And once we have this significance threshold, basically all windows that have a p-value less than this threshold are significant. Okay, so we use some real sequencing data like whole exome and whole genome data to derive this, to calculate these thresholds. So for whole exome data, we use data on 4,000 individuals from an autism sequencing study. For whole genome data, we use data on 1,000 whole genomes from the Simons simplex collection. And we employed the sliding window approach. Of course, we don't know what the, in general, we don't know what the correct window size is, so we try window sizes between 5 kb and 50 kb. Okay? And so these are the thresholds that we get. So if you have exome data, and instead of doing a gene-based analysis, you do the sliding window approach. For exome, we get a threshold of 1.4 times 10 to minus 6. So this is very close to the threshold we normally use for whole exome data when we adjust for 20,000 genes. For whole genome, we get a threshold of 4.8 times 10 to minus 8. So this is again similar to the threshold we use for in GWAS studies. And then we can also get, study-specific thresholds. So I mentioned the Metabochip study where we looked at 100 regions. So for that study, the threshold is 1 times 10 to minus 5. And if we incorporate function annotation, so we have, instead of 1 test per window, we now have 128 tests per window, then the threshold is a little bit more stringent, but it's not as stringent as it would be if you did like Bonferroni adjustment. Okay, so now going back to the Metabochip analysis I showed before. So the top figure is simply the gene-based analysis I already showed. The bottom figure shows, the scan statistic approach where we ignore the gene information and we just, use the sliding window approach with windows between 5 and 50 kb. And so I'm showing here the windows that are, above the threshold. So what you probably can easily see is that we are able to identify more significant windows by doing the sliding window approach versus a gene-based approach. And this has to do with the fact that, oftentimes this, the signal clusters within, regions of a gene, and so this type of sliding window approach is more sensitive to that scenario than, just doing a gene-based approach. Okay? So I'm going to now talk about so I talked about how we integrate these functional annotations into the testing procedure. So I'm going to talk a little bit about how we derive those functional scores. So what do we mean by functional variant? And there are different ways to define functional variants. So of course we have the experimental approach that evaluates the effect of a sequence change on a phenotype. This approach is nowadays have modest throughput, although as you all know there is a lot of effort in developing high throughput assays for functional assays like CRISPR-Cas9 and multiplex reporter assays and so on. And then there are other more computational approaches like evolutionary approach that quantifies the extent of selective constraint at a position or in a region. And then there is a biochemical approach that measures evidence of molecular activity across the genome. And so roadmap epigenomics is a large-scale project that employed this biochemical approach. So they basically performed biochemical assays genome-wide across a large number of tissues and cell types in the human body. And so basically at the end of the day, if we look at one particular region, what we have measurements from these different assays, so histone modification assays, open chromatin assays. And so what we want to do is integrate these different types of data to predict the functional effects of genetic variants. Okay? So one of the first methods we proposed for doing this integrative analysis is called Eigen, and it's a spectral approach to to integrate many different functional annotations. So Eigen assesses functional effect at the organism level, so it gives you a score for a variant, how functional that variant is likely to be. Okay? Now, suppose you're studying a particular trait like, say, autism. We're mostly interested in, whether variants are functional in brain or in particular regions of the brain. So instead of having this one single score per variant, we like to basically be able to predict functional effects across different tissues and cell types. Okay, so we have recently proposed this method called FUN-LDA that does prediction of functional effect at the level of the tissue or cell type. It's an unsupervised approach, and it's based on a model called latent Dirichlet allocation model. So this model is very popular in the topic modeling literature, and it's also been used in genetics before. It's a model that underlies a population admixture algorithm called structure. Okay? So the underlying idea, basically, we assume we have biochemical assay data for 127 tissues and cell types from roadmap and ENCODE. So for each variant I, or position I, and tissue K, we have a vector of functional scores. This histone modification, open chromatin scores, and so on. And basically, we want to fit an LDA model. So the main assumptions are we assume that these tissue-specific scores are modeled as a mixture over latent function classes. So these latent function classes are, enhancer, promoter, active transcription, and so on. We assume that the mixture components are shared across all the tissues, and the mixture proportions can vary from tissue to tissue. So basically, in more details, we assume basically that the function annotation data for tissue K has been generated from the following generative process. We let alpha be the hyperparameter vector. Then for each tissue K, we choose a distribution over functional classes. So pi K from this Dirichlet distribution. Then for each variant I in tissue K, we choose a class. And given the class, basically, we generate the functional annotation data from some random distribution F. And our goal is basically to calculate the probabilities that the variant is functional in a function class C in tissue K given Z and alpha. Okay? So, to fit that model, we use a variational inference algorithm that I will not go over here. So some of the advantages of the FUN-LDA approach, well, it allows for multiple functional classes. So in the original Eigen approach that we proposed, we only allowed for 2 classes, functional and non-functional. It can incorporate annotations with arbitrary distributions, so you don't really need to do any pre-processing. You don't have to, do peak calling or binarize your data to use this algorithm. And of course, we can get approximate probabilities for variants to be functional in each tissue or cell type separately. So the type of annotations I've been using in the Metabochip data are really from this FUN-LDA approach, these probabilities for variants to be functional in different tissues. And we have a website that where you can score variants of interest, or you can download all the scores if you're interested. So I want to, in the time I have left, I want to talk about an extension we've developed recently to the case where we have some labeled data. So as I mentioned, the FUN-LDA is a purely unsupervised approach. It doesn't make use of any labeled information. But suppose that, for certain cell types where you might have a small set of variants that are labeled, right? From, say, an experiment. You've run an experiment in a particular cell type, and you've identified, you tested a set of variants, say from CRISPR-Cas9 or multiplex reporter assay. So how can we incorporate that type of labeled information into our prediction to improve the prediction accuracy over a purely unsupervised approach? So we developed this semi-supervised approach called GenoNet. It takes as input a set of labeled variants, M. So for these variants, we have a binary label, 1 or 0. And then we have a large number of unlabeled variants. So we don't, unlabeled in the sense that we don't have experimental labels. But of course, we have predictions from some unsupervised approach, say FUN-LDA approach. And then we have function annotation data on a large number of, a large number of features. So in this particular implementation, we used over 1,000 different features. And all this data is going into the GenoNet algorithm, and then we're going to get GenoNet prediction for every position genome-wide. So here's basic ideas behind GenoNet. So as I said, we assume we have M variants for which we have experimental labels. And then we have L-M variants for which we have, we don't have experimental labels, but we have, FUN-LDA predictions. And then we have a set of features. Okay, so M is the number of labeled variants is small. In fact, for many cell types, we don't have any labeled variants. Then L is L can be very large, so the total number of variants, and then we have thousands of features. And so what we're interested in is basically estimating this prediction function by solving this optimization problem. So the objective function has two components. One is for the for the labeled part, and this is a penalized log likelihood with an L1 and L2 penalty, like in elastic net. And then we have a second component, which is for the unsupervised part. So this is like this sum of squared differences between the predictions from say FUN-LDA and the values predicted by the prediction function f. Okay, and gamma L is a tuning parameter that controls how the unlabeled data is being used. So when gamma L is 0, we get the fully supervised approach. When gamma L is infinity, we get the fully unsupervised approach. And then, gamma L in between, we get the semi-supervised approach. Okay, and so one of the nice properties of GenoNet is that you can actually compute these scores even for cell types or tissue types for which we don't have experimentally confirmed variants. And in that case, basically, when M is 0, we basically the predictions from GenoNet are just linear approximations of the FUN-LDA posterior probabilities. So I'm going to skip how we actually do the estimation, and I'm going to show you some results. So one question is, how does a semi-supervised approach compares with a fully supervised approach or a fully unsupervised approach? And we looked at two factors that affect the performance of the supervised methods, the number of labeled variants that's available in the training, and the amount of label misspecification. So what I'm showing here on the left is the accuracy of the supervised, semi-supervised, and the unsupervised approach as you increase the number of labeled variants. So when the number of labeled variants is small, basically the supervised approach, so it's shown in red, is doing worse than a fully unsupervised approach like FUN-LDA. As the number of labeled variants increases, then you get that the supervised approach is better than the unsupervised approach. And you see that the semi-supervised approach, shown in blue, is actually doing pretty well across the different scenarios here. And it's kind of a similar behavior if you look at label misspecification. So when the label misspecification is high, you have the unsupervised approaches are more accurate than the supervised approaches, and as the label misspecification decreases, then you obviously get the supervised approaches doing better than the unsupervised. So that's something to bear in mind, because at this point in time we are kind of towards the left part of these plots where we have low number of labeled variants and not great accuracy. So in those cases, this unsupervised or semi-supervised approaches actually can work better. Well, so we compared the accuracy of GenoNet with existing methods, and I'm going to show here a comparison on data from 3 cell lines where we actually had some MPRA validated variants. And we compared with different type of methods, so tissue specific, like I mentioned FUN-LDA. We also compared with GenoSkyline, which is like another mixture model with only 2 components. And also just plain quantitative DNAs. And then we compared with some organism level prediction methods like Eigen, SKAT, which is very popular, Linsight, and so on. And we compare these methods in terms of 3 different criteria. So we compared with respect to AUROC, so area under the receiver operating curve, AUPR, the area under the precision recall curve, and also in terms of correlation between the predicted labels and the true labels. So these are the results for the 3 different cell lines. So on the x-axis is AUPR, on the y-axis is AUROC, and then the size of the bubble is basically relates to the correlation between the predicted and the true labels. So our method, GenoNet, is shown in blue, and overall, basically, with respect to all these 3 criteria, GenoNet performs better than these existing methods. And I'm going to end with some application of this function square. So I talked about in the beginning about the first application of incorporating those function square into the gene-based association or region-based association test. I'm going to show now an application to find mapping. So we basically selected locus from GWAS on schizophrenia from the PGC 2014 study, and we focus on a particular locus where this microRNA gene is located, because there is some functional follow-up that's been done in this paper at this locus. So basically, they did some ataxic experiments in human-induced pluripotent stem cell-derived neurons, and they used some other information on transcription factor binding sites to prioritize one SNP. And then they use CRISPR-Cas9 editing to show that this variant is functional and affects the expression of this gene, and has some other functional effects. So what we wanted to see is whether by using these computational predictions we have from GenoNet, whether we can correctly guess where the functional variants are located in this region. So what's shown here, the blue SNP is the GWAS index SNP. So this is a SNP with the smallest p-value in the GWAS study. And then there are a bunch of SNPs shown in gray here that have similar p-values. So the p-value is shown here on the y-axis, on this y-axis. So these are all highly correlated with the blue SNP. And then the red SNP is the one where they've done functional, CRISPR validation. Okay? And so what's shown on the first y-axis is a GenoNet score in these H1-derived neuronal progenitor culture cells. So what you see is that where this red SNP is located, we also get a high functional score from GenoNet. We also get high functional score in this region, and it turns out that where this green variant is a known risk variant. It's a rare variant, but it's a risk variant for schizophrenia. So basically, by using this functional score, we show that we can, at least in this example, narrow down the list of variants of potential candidates for follow-up, and luckily in this example, it includes a variant that was validated using CRISPR. So how much time do I have?
1 more.
Okay. Yeah, so I think we've heard, yesterday about LD square regression. So you can actually use these functional squares across different tissues to combine them with a LD square regression tool to estimate the contribution of heritability that's contributed by variants that are functional in a particular tissue. Okay, and so using summary statistics across a number of GWAS studies, we've run this LD square regression using our functional predictions from FUN-LDA. And what I'm showing here for each trait, in the first column I'm showing the top roadmap tissues that came up in the LD square regression approach, and basically you get expected results. For schizophrenia we get the most significant tissue is brain. For BMI we also get brain. For these lipid traits we get liver. Type 2 diabetes we get pancreatic islets and so on. So so overall, basically, what I want to say is that these functional predictions across different tissues, they have very useful applications in complex trait genetics. To summarize, I first discussed statistical tests for whole genome data and how we can integrate different function annotations to improve power of those tests. And then I talk about unsupervised and semi-supervised approaches for predicting functional effects in a tissue or cell type specific manner. And I like to thank people in my group who have worked on this project, in particular Zihuai, Daniel and Ken, and some other collaborators. Thank you.
Yeah, time for a couple of questions.
Yes.
Can you clarify when you do like time mapping or the testing, are you picking one of the 127 cell tissue types? Are you doing some joint analysis with all of them together, and you're just picking one? How do you pick it?
Right. Yeah, so in that example, we just do it tissue by tissue or cell type by cell type. So I just showed you one that we thought was, similar to the tissue where they did the functional study. But in fact, it's actually, this variant turns out not to be, so tissue specific. So it's functional in a bunch of other different cell types.
So would you mind detail a little bit? I lost you when you spoke about this sliding window, and what is the connection between this and the extreme value distribution, and how you develop that function there?
Well, it's so when you do the sliding window, right? You slide the window of, say, a fixed size W across the region, and then your scan statistic is going to be, the max of the overall windows of the negative log p-value. So that's an extreme value statistic, right? And so we can, what we're interested in is basically getting the quantiles of the distribution for that statistic, right? To be able to estimate the threshold. Right? So anyway, so this negative log p-value, it's distributed as an exponential with rate 1, and then basically we can use the results from the Gumbel distribution to estimate these quantiles. But we have to do some resampling to take into resampling to basically take into account the correlation among the different windows to estimate the moments of that distribution, basically.
You estimated the distribution of T in the two data sets in the later slides.
Right.
Yeah. So how did you do that? Like, did you just calculate the T for each of the windows and then use that as a numerical, not very.
Yeah, so basically, I didn't really explain this very well in detail, but yeah, we have basically, so basically the reason it depends on these individual studies is because we assess the correlation empirically among the test statistics in different windows. So for each, so these thresholds that we derive from, say applying the sliding window approach to the different data sets, so they depend on the correlation among the test statistics, and that correlation we evaluate empirically from, using this data.
If I want to.
Maybe we can discuss it during coffee because we are way over time.